Today, I chose to do my Chapter 3 mini lecture on the last part of the chapter, which is about how we study the brain, how psychologists study the brain. So neuroscientists are like mechanics as they try to figure out what's going on in the brain, when it works improperly, and most importantly, how. Neuroscientists are like mechanics in that they have to figure out how things work, but mechanics differ because mechanics can look up detailed descriptions of the car that they're fixing or whatever, and neuroscientists can't exactly look up detailed descriptions of the brain. So neuroscientists are essentially and quite literally figuring out themselves how the brain works as they're trying to figure it out. So the first way that we can map, or psychologists and neuroscientists can map brain function through lesions and electrical stimulation. And before the 20th century, the only way to map out brain function was the lesion method. So just a little vocab. A lesion is an area of tissue damage, whether it be in the brain, in the rest of the body, whatever. And then the transcranial magnetic stimulation, or TMS, is the temporary localized disruption of the functioning of a brain region produced by the application of strong magnetic fields or coils. And I'll talk more about that in a bit. So, as I said, before the 20th century, the only way to map out brain function was the lesion method, and the only way to study the brain was to physically remove it from a body after that patient had died. The lobotomy is the deliberate cutting of connections in the frontal lobe, and that was also a use of lesioning. So TMS, or as I said before, transcranial magnetic stimulation, is essentially using coils placed over the skull and the scalp and disrupting brain function under that portion of the skull for a period of time. So it's currently being tested as treatments for things like depression, and essentially it's used to experimentally locate brain regions that influence specific behaviors. So most lesions and TMS experiments use non-human subjects, but the results can often be the same between humans and animals. A couple points to this. Basically, stimulating the left frontal cortex will cause movement on the right side of the body. That's a connection between animals and humans. Stroking the skin on the right side of the body will cause neurons in the left parietal lobe to fire action potentials. That's also a connection between humans and animals that they found. And then lesioning the left occipital lobe will disrupt vision on the right. That's also another connection. So the next section is about scientists eavesdropping on electrical signals within the brain. Now, that sounds a little strange, but essentially the way they do this is through a electroencephalogram, or an EEG. I hope I said that right. So an EEG is a recording of fluctuating potentials taking place in the brain, and it's made by an encephalograph. And then we also have mirror neurons, which are neurons that are both active when an individual makes a particular movement, and then when that individual sees another individual making that same movement. So that's, in the textbook, there's kind of a visual about a monkey and picking up raisins, watching a human pick up raisins, and then that mirror neuron response. So going back to the electroencephalogram, or EEG, it's used for neuroscientists to essentially eavesdrop on the brain, like I said before, and it reveals electrical activity. In animal subjects, neuroscientists can see similar workings of the human brain. So this is interesting because it can locate, by comparing EEG signals from many different pairs of wires on the scalp, it can be used to locate where a seizure begins and then to monitor its spread, and that's obviously very helpful. So the next portion of this section is about today's imaging technology, and obviously this is very advanced compared to before the 20th century and everything like that. So today's imaging technology can reveal the brain's interior structure and function. So the vocabulary of this is also kind of the summary of this portion of the chapter because it explains all the different types of tests. So the first one is the computed tomography, or CT, and that's a technique for examining brain structure through computer analysis of X-ray particles passing through the head from several different positions. So essentially an X-ray just sends through a fluid-filled space and then seeing how many X-ray particles come out the different side. And then doing that in a 3D formation will lead to the CT. The next is the magnetic resonance imaging, and I'm sure everybody knows that's an MRI. So that's a technique that uses magnetic fields to generate images revealing structural details in the living brain. Next is a position emission tomography, or a PET or PET, kind of like a PET scan. That's a technique for examining brain function using injections of radioactive substances used by the brain, and then particle detectors and computers are used to find where most of the radioactive markers gather in the brain. If the radioactive marker is part of a chemical that binds to a particular neurotransmitter receptors, PET or PET can even reveal how those receptors are distributed in the brain, which is interesting as well. And then the final vocabulary term, or kind of point of this section, is the functional MRI, or fMRI. So this is magnetic resonance imaging that detects changes in blood flow and oxygen content and identifies regions of the brain that are particularly active during a given task. So that's interesting to note because you can see the fMRI can detect things like which brain regions are active when you're looking at colors or moving a finger or even reading a book or making dinner or things like that. So in general, this is kind of obvious, but all the above are ways to see inside the brain without looking, like actually physically looking at a brain. And this is interesting because, like I mentioned previously, before the 20th century, the only way to actually see inside the brain was after a patient died and physically removing the brain. So the next section, it's kind of short, is we infer brain function by comparing different mental activities. I'm just going to summarize this, but the entire brain is active when we're awake, even if we're completing a boring action like looking at a dot or staring at something. And this just goes to prove that the notion that we only use 10% of our brain is very much false, because we use our entire brain, whether even when we're awake, no matter what we're doing. So the question is, which parts of the brain are more active depending on which task we're completing, because there are different tasks for different regions of the brain. So each region of the brain has essentially a different task. Finally, I'm going to talk about how psychology and biology use two different vocabularies to describe the same thing. So vocabulary for this is plasticity, which is also called neuroplasticity, and it refers to the ability of the brain to change in structure or function, which is pretty interesting and one of the reasons I chose this section. So essentially, your brain is constantly changing and taking in new information. For example, you remember the nice thing that happened last week or something great that happened recently because your brain altered its structure to allow you to remember it. So the mind uses this and essentially remembers what it wants to remembers the nice things, and just makes way for the brain to change and alter itself physically for those good things that happen. So that's basically it. There's a section inside this last section, I guess you could say, about mapping the connectome in the human brain. And this includes diffusion tensor imaging, or DTI, which is a brain imaging technique that reveals axonal connections. And that's pretty interesting as well. So that's it for my section of this chapter, chapter 3, about how psychologists and neuroscientists study the brain.